Buongiorno. I'm just going to tell you a little bit of our story of how we were implementing the Watchmaker Library Preps at the Crick. So I just moved to Milan to the EO, and I'm probably going to touch many of the points that Jen talked about this morning. So this is the Francis Crick. I was their project manager for 3.5 years until March this year, and that was our lab and the team at that point. And at the genomics facility at the Crick, we work with only Crick groups, and we work with about 100 groups that do genomics, and about 400 projects a year. We sequence around 14 to 15,000 samples a year. We have a team dedicated for automation. We do library prep. We do extraction of nucleic acids. We have single cell, spatial transcriptomics, all genomics that you can think of. And as Jen was pointing out, when I joined as a project manager, we were deciding which library prep kit we would use per sample. So whenever we receive a sample or samples from a project, we would look at the quality and the input quantity and decide which library prep we would use. So we had the main library prep kit, which was poly-A based for the samples that were good quality and high concentration. Then for those samples that had lower quality or lower concentration, we would move for ribosomal depletion. Then we had a different kit for low input, and then we had a highly specialized, really expensive kit for samples that were either really lowly concentrated or highly degraded or FFPE. So Watchmaker kit actually covers the majority, I would say 99% of the samples that we did at the Crick at that point. And that's why we ended up changing. And we changed, I will just tell you three little stories about how we decided to change. So everything started with the Crick African Network. So at the Crick, there is an effort to give back some of what we get. So every year there is some fellowships that are given to African researchers so they can come to the Crick and learn new technologies to bring back to their countries. So we had Kani coming from Africa, and she brought some human samples from a cohort of kids from Côte d'Ivoire that had a upper respiratory tract infection, and they followed these kids through time. So they collected blood and mouth swabs from these kids through the time, and she brought the samples back to the Crick and extracted the RNA from them. And for her, it was about 200 samples, and she wanted to do all of the libraries made with the same kit, and she wanted to be able to compare all of these samples across time. So when she extracted the RNA, we saw that there was a problem, basically, because the blood samples, they had good quality and high quantity, but also they contained globin, and they had not been DNA treated. And then for the mouth swab samples, we got low RIN samples and low concentration. So we were thinking which kind of kit we could use for this, and we decided, because also the money was constrained, because it was more than 200 samples, we would move to one of these kits that is a 3' low-cost kit to perform these libraries. So we tried with a few of the samples, and the first thing we saw is that the libraries look awkward. So our usual RNA-seq library prep should look like this, and just a normal size distribution centered around 200 to 300 base pairs. But the samples from this project look banded, which was really strange. We did low-pass sequencing of these samples, and first thing we can observe is that for even less than 10 million reads, the duplication rates for these libraries were massive, more than 90%. And then the mapping to the human genome for the blood sample was okay, but for the swab samples was really low. So we were not happy with this. We contacted colleagues at different facilities across UK. We especially contacted people in Oxford because we knew that they were using this kit, and they were telling us that it's a tricky kit, that it's really susceptible to ambient temperature. So we tried repeating these libraries, but we had no luck. We heard from our colleagues at at UCL that they were testing this kit from this new company that we've never heard about. So we went to a conference, and I met their Amy, and I explained to her our problem, and she said, "Oh, no problem. I'm sure our kit will be amazing. It will work. So we will provide you some kits, and you can try." So we tried with the Watchmaker Polaris Depletion, and the first thing you can see is that the libraries that we made, they look normal. So we were really happy to see that. And then when we looked at the sequencing results, so this time we sequenced to higher coverage, and what we saw is the duplication rates for the blood samples decreased a lot, and the mapping to human genome increased to almost 100%. But we were surprised because our swab samples kind of look awkward, so there was still a lot of high duplication rates, and the mappability to the human genome decreased. So we were really puzzled. We contacted Watchmaker, and we had a chat with their R&D team, and they said, okay, please give us the FASTQ files. We will have a look and see what's going on. They looked at our FASTQ files, and they saw that the majority of the reads that were not mapping to the human genome were actually ribosomal RNA from bacteria, which makes sense because these were kids that had an upper respiratory tract infection, so we were basically swabbing bacteria from them. So with the Watchmaker team, we talked to them, and they said, okay, what we can do is, together with the Polaris Depletion, we can design some probes that you can add to deplete for the bacterial ribosomal RNA. So we spoke with them, and I told them, just give us some probes for these genes, for these species that we see in the samples, but also since we have to order these probes, could you also design some probes for normal commensal bacteria for the upper respiratory tract so we can use this for other projects? So they were really helpful. They designed the probes, we ordered them, now we have probes for the rest of our lives because we have like 100 mLs of probes, and you only use 1 microliter per sample. We've used it already for other samples, so it was great. And we were actually, when we used these probes, we saw that we were able to improve the mappability to the human genome. So for some of these samples where we were having less than 10% of human reads, we were able to increase to over 50% of human reads. So we were able to rescue this project. When we saw, so we repeated the experiment, and we used also these bacterial probes for the blood because we wanted to keep it consistent across both types of samples. And also because we saw that the library preps from Watchmaker were being over-amplified, we reduced the PCR cycles, and we were able to decrease the percentage of duplication rates in our blood samples, so we increased the quality. And then for the majority of the swab samples, we were able to increase the quality. So we were able to get a higher percentage of mappability to the human genome. So some of the samples still had some bacterial contamination, but that's okay. And the duplication rates were sort of fine. So basically from this little story, we can tell you that the 3' kit displayed too many read duplicates and it was inconsistent, so we discarded it. The Watchmaker Polaris Depletion reduced the number of duplicated reads in the challenging samples. And we were really happy with the engagement from the research and development team that helped us succeed with this project. All of this work was done by Emma at the Crick. And because I was starting to be really happy with this kit, also the team was super excited that the library prep is super easy. It takes only 5 hours. The protocol is not complicated. So we decided let's give it a go to see other challenging library types that we have. So one of these is the FFPE samples. So this is just a quick overview of an experiment that is done at the Crick by Ciccarelli Lab. So they work on colorectal cancer, and we had done for them hundreds of samples from laser capture microdissection from FFPE blocks. So they were separating tumor and stroma cells, extracting RNA, and then we were using this highly specialized kit for RNA library prep that cost over £200 per sample. So I told them, we have this kit, do you mind if we try with your samples and see if it will work, because the protocol is much more streamlined. This other kit takes about 3 days to prepare the library. Do you mind if we try? So they said, oh yes, give it a go. So they gave us the worst sample that they have. And Watchmaker recommends starting from 10 nanograms for FFPE samples. Here we started with 1 nanogram, and we were able to see, so this is the same samples, prepared with the specialized library prep kit, so you can see that the percentage of aligned reads to the human genome is quite low, whereas with the Watchmaker kit starting with 10 times less material than Watchmaker recommends, we were able to map over 75% of the reads. And also we were able to recover a higher number of genes, sorry. And for these highly specialized kits, we were using twice as much RNA to begin with. So again, if you look at our library prep decision framework, we saw that Watchmaker would be able to cover the majority of our samples that we prep, and our preferred input at the Crick was 100 nanograms, so we would be able to substitute almost everything. So if we were to change, sometimes they say if it ain't broken, don't fix it, but this kit is so much faster. The number of PCR cycles is lower. So we decided let's try with other samples to see if we can swap all of our library prep kits to this one. So for choosing a new kit, we would want it to have a high range of inputs and a high range of qualities. So we decided to test it with real samples from another lab. So these come from renal tumors from the Turjalik lab, and we had samples from this patient where we got RNA extracted from a renal tumor, RNA extracted from the normal tissue, which had a medium to high RIN, and then some organoids derived from the tumor that have a high RIN. We did a rather convoluted experiment from which I'm just going to talk to you about the 100 nanograms, but we were trying to see what was the minimum input qualities and quantities that we could use with this kit. So we tried our standard kit, which was with poly-A enrichment, and our standard kit with the ribosomal depletion, and we tested the poly-A from Watchmaker and the Polaris Depletion. And the first thing that is really noticeable is when you look at with the Polaris Depletion, same starting material, the library prep from Watchmaker yields so much higher DNA concentration at the end, so much higher library prep, even though we had less PCR cycles. So for the other kit, we did 11 PCR cycles, and we got around 10, 20 nanomolar libraries, whereas with Watchmaker, we did 9 PCR cycles, and as you see, we had too much library, almost 90, 80 nanomolar, in some cases 100 nanomolar library concentration. And the percent, even though we had so much more library, the percentage of duplicated reads was even lower. So we decided, if we are going to keep using this kit, we told Watchmaker, I think you need to change your protocol. You need to reduce the library, the number of PCR cycles, and we did this ever since. When we look at the samples overall from all the different kits that we use, we see that the samples separate. When you look at the clustering, the samples separate by origin, so the samples that were coming from the tumor all clustered together, the samples that came from the healthy tissue clustered together, and the samples that came from derived from the organoids, they all clustered together. And then when you look at the other component, you see that they cluster by enrichment type. So the samples that were prepared with poly-A from both kits look exactly the same, and then the samples that were prepared with ribosomal depletion look slightly different because the ribosomal depletion strategies are different. Then when we compare the poly-A library prep kits, we saw that there's basically no difference. So we were happy that we could just swap both kits, and no researcher would know the difference between them. So there was about 24 upregulated genes and 10 downregulated genes, but this is just due to a stochastic and the way differential gene expression with DSEQ works. One thing that is important to show, I think, just to remind people, is when you do poly-A enrichment, you are enriching through the 3' end of the kits. So if you have, we call it medium quality. It was actually quite high quality. It was 7.9 RIN, but still you can see that with either kit that you use, there is, if you do poly-A enrichment, you see a 3' bias of your library, whereas if you do ribosomal depletion, you see a stronger coverage across the whole gene body. So depending on what's your application, you might want to choose one or the other kit, just something to have in mind. Whereas with the high-quality samples, you see that the coverage is consistent across all library prep kits. And one thing that Jen pointed out before, so we wanted to know also for this project, does it make a big difference if we start from 500 nanograms or if we start from 0.5 nanograms. So again, Watchmaker doesn't recommend to go so low, but we just tried just to see how far we could push the kids. And we see that even starting with the 500 versus 100 nanogram doesn't make much of a difference between the inputs. So we can work with less material, have more material for other types of analysis. And even started with 0.5 nanograms works nicely. So this was evaluated by these two of our team members, and the take-home messages is that both library prep kits that we're using produce good libraries. Polaris Depletion works with even as low inputs as 0.5 nanograms. This library prep kit is highly efficient. You can reduce the number of PCR cycles, and the results from both kits for poly-A enrichment are the same, so we were able to substitute both kits happily. And the differences that we saw in differential gene expression between the same input prepared with both library prep types basically is due to lowly expressed genes. So because we saw that Watchmaker Library Prep is flexible, the comparable results with the previous kits that we were using, but is also faster, and it allows for higher input RIN and concentration, we decided to change. And also we are really happy with their customer support. So this was the team, and thank you for listening.